This video is the second in a series about nucleotides for OCR A-level biology. DNA replication is often described as semi-conservative. This means that in each new DNA molecule, half of the DNA has been kept from the previous old molecule and half is brand new made from fresh nucleotides. When DNA is replicated, the hydrogen bonds between the complementary bases break down, and then the two strands are separated. A new complementary strand is then synthesized against each of the original strands, making two semi-conservatively replicated DNA strands. When somatic cells, so all of the cells in an organism apart from the gametes, divide, they do so by mitosis, and it's important that each cell replicates its DNA exactly. This allows each of the new daughter cells to have the exact same DNA as the parent cell. It's possible for this to be achieved because of the complementary base pairing between the two strands of DNA. If we can compare them, we see that guanine is always paired with cytosine and adenine is always paired with thymine. This semi-conservative DNA replication happens during the S phase of interphase in the cell cycle. In the previous video, we learned how the complementary base pairs in DNA and RNA bond together. So pause the video and see whether you can write down the complementary DNA and RNA sequences for this DNA sequence. Hopefully you've managed to do this successfully. Double check your work now. Make sure that where you're thinking of the RNA sequence, you don't have any thymines and instead these have been replaced by uracil. The first step in semi-conservative DNA replication is for the DNA to be unzipped. This is done by an enzyme called DNA helicase, which is responsible for breaking the hydrogen bonds between guanine and cytosine and between adenine and thymine. A simple 2-mark exam question might ask you to describe the role of DNA helicase. So we'd talk about how it unzips the double-stranded DNA by breaking the hydrogen bonds between complementary bases. Now that the two parent strands of DNA have been separated, free-floating nucleotides are used to make a new complementary strand. These are joined together by another enzyme called DNA polymerase. The new strand of DNA will be anti-parallel to the original strand, so the 5' end of each strand is at opposite ends. Following this process, we have two identical daughter molecules. So to summarise so far, DNA helicase breaks the hydrogen bonds between the bases, and this causes the DNA to unzip, exposing those bases. Complementary nucleotides then line up alongside both of the original strands, and DNA polymerase forms new phosphodiester bonds, joining those nucleotides together. The hydrogen bonds will then naturally reform between the complementary strands, and each new DNA molecule contains half of the original DNA molecule. When you picture DNA replication, it can be easy to imagine that the entire DNA molecule unzips in one go. But remember that that DNA molecule contains hundreds of millions of base pairs, so that would be quite impractical. So what happens instead is that the DNA unzips gradually bit by bit, creating what's called a replication fork. Now this means that the process of synthesizing the new DNA is actually a little bit more complicated, because the enzymes that are involved can't always get to the bit of DNA they need. And this means we have two processes, continuous and discontinuous DNA replication. As you know, the strands of DNA run anti-parallel to one another. So if you look at my top strand, it's running 5' to 3', and at the bottom we have 3' to 5'. And the newly synthesized DNA against each of those will be in the opposite direction again. Now, DNA polymerase always works 5' to 3'. But on the lower strand, you can see that that's going to be quite challenging. So on my upper strand, DNA polymerase will attach here at the 5' end, and it will just synthesize in that direction. This is what we call continuous replication. So the DNA polymerase will move from 5' to 3' of the new strand, and it will create a strand that we call the leading strand. So this is what's made by continuous replication, and as the DNA carries on unzipping, it can just keep making that leading strand longer and longer. In contrast to that, at the bottom of the screen, you can see that it's not possible for the DNA polymerase to go right to the 5' end of the DNA, because it's not open yet. So what it does instead is it attaches where it can in the replication fork, and it synthesizes 5' to 3', and produces what we call an Okazaki fragment. As more of the DNA unzips, the DNA polymerase reattaches further along 5' and produces another Okazaki fragment. A third enzyme, called DNA ligase, can then join these Okazaki fragments together in order to produce the lagging strand. This is called discontinuous replication. Pause the video and see whether you can describe the difference between continuous and discontinuous replication. Continuous replication is going to take place on the leading strand. It happens in the same direction that the replication fork is unwinding, and DNA polymerase adds nucleotides in one long chain. Discontinuous replication takes place on the lagging strand. DNA polymerase works in the opposite direction to the replication fork unwinding, and then these short Okazaki fragments are later joined together by DNA ligase. You already know from your GCSE studies that mutation is a change in the DNA sequence, like this adenine here turning into guanine. This may be spontaneous or random, or due to exposure to a carcinogen like radiation, toxic chemicals, or UV. But you may not be aware how many of these mutations happen during DNA replication. If the mismatch repair enzymes within the cell don't identify that an incorrect nucleotide has been added in during semi-conservative DNA replication, the mutation can cause permanent changes, which can lead to conditions such as cancer. However, not every change in the DNA sequence will lead to problems for the organism, and this is because the DNA code is degenerate or redundant. What this means is that there are several codons which all code for the same amino acid. For instance, these first 4 all code for glycine, and that means that if the third nucleotide of that triplet codon is mutated, it's not going to affect the sequence of the amino acid. Likewise, we've got 2 for aspartic acid and 2 for glutamic acid. Time for another progress check. What name is given to the process of spontaneous change in DNA? What type of diseases can this lead to? And what are the roles of DNA helicase and DNA polymerase in semi-conservative DNA replication? These spontaneous changes are called mutation, and they can lead to diseases such as cancer. DNA helicase is responsible for unzipping the DNA, breaking the hydrogen bonds, whereas DNA polymerase works 5' to 3', forming new phosphodiester bonds between nucleotides. Thank you very much for watching, and I hope you found this a useful summary of semi-conservative DNA replication. If you did find this video useful, then don't forget to like, subscribe, comment and share the channel, and I'll be back soon with some more A-level biology.